I guess now we will welcome our second speaker, Valentina Venzin from Pasteur Institute in France. The title of her talk today is CD4 T cells license cover cells to produce IL-27 and reverse CD8 T cells dysfunction induced by hepatocellular priming. Welcome, Valentina.
Hello, everyone. Really happy to be here in this wonderful initiative. Um, I'm Valentina. I'm a postdoc. I recently joined Yasmin Belkait's lab at the Institut Pasteur in Paris, but my PhD roots are actually in Matteo Iannacone's lab at the University of San Raffaele in Milan. And during my PhD, we together try to understand the dynamic of T cell cooperation inside the liver. So Matteo's lab is interested and focused in defining how immune responses are built in peripheral organs with a particular focus on the liver. And in Matteo's lab, we used HBV pathogenesis as a model to dissect how immune responses are built inside this organ. And HBV provides a really nice model to study this because of many reasons. So first of all, is a virus with a super strict tropism only for hepatocytes. And in these cells, it replicates in a non-cytopathic way. So this basically means that during the initial phases of the immune response against the virus, there's no really a strong innate immune response. And this renders the adaptive immune responses the main responsible for viral clearance. And in particular, CD8s have been found to be the more crucial cells responsible for the killing of the infected hepatocytes. However, what we know about hepatitis B virus pathogenesis is that unfortunately, during the chronic, this virus can cause both acute and chronic disease. And during the chronic disease, CD8s are dysfunctional. We still don't know really precisely why these CD8s are dysfunctional in this context, but Matteo's lab has done beautiful work defining one of the possible mechanisms that can explain this dysfunctionality during hepatic HBV chronic infection, and is a process that we refer to as hepatocellular priming. So basically, a really peculiar immunological event can happen during HBV infection, and it's that basically during HBV infection, CD8 T cells can be primed directly inside the liver. And this is kind of weird because this is not the place where usually CD8s are primed. When this happens, CD8s are primed by infected hepatocytes, which are not professional antigen presenting cells. And this basically leads to the establishment of a dysfunctional phenotype in CD8 T cells. And basically, briefly, they can proliferate locally in the liver, but they won't be able to become effectors and kill the infected cells in the liver. Now, a lot of effort has been done in the field in order to understand how to reinvigorate these CD8s. And one feasible way to do this, for instance, is finding new modulators or way to locally activate professional antigen-presenting cells inside the liver so that these cells can cross-present correctly and correctively CD8 T cells inside the liver, leading to the reinvigoration of their response. So this was the starting point of my project in Matteo's lab. We started with a very simple question. What about CD4 T cells? Because we know CD4s are main orchestrator of CD8 T cells antiviral immunity, and they usually orchestrate CD8s via antigen-presenting cells, so modulating the function of antigen-presenting cells. So we started this project with a question, understanding to which extent the priming and function of CD8 T cells inside the liver, in the context of HB pathogenesis, relies on CD4 T cell health. So first thing first, we generate a new mouse model that we called M126. These mice, very briefly, all CD4s are specific for HBV, and we use these mice as donors of CD4s. Now, what we do in our working model, we take CD4s from these mice and we differentiate them in effector CD4s with a Th1-like phenotype. So basically, effector CD4s specific for HBV and ready to help. And we put these CD4s in our working model. So HBV doesn't infect mice, but we have a model to resemble HBV pathogenesis. Basically, these are HBV transgenic mice, are recipient mice in which the HBV virus replicates in the hepatocytes, only in hepatocytes with no evidence of cytopathology. If we transfer in these mice naive CD8 specific for the virus, these become dysfunctional. So it happens what exactly I just showed you and explained to you. So basically, CD8s won't differentiate into effectors, and we will have a kind of a chronic response of these against the virus. So we wonder, okay, what maybe CD4s in this context, if we add some effector CD4s, can ameliorate the CD8 T cell response? And well, this is what happened, because if we put in this system now CD4s, and is the yellow and blue combined dot, you can see that we have some liver damage that we measure at transaminase level. So basically, some hepatocytes are dying. Effector CD8 T cells are more in number, they produce more interferon gamma, they proliferate more, and in general, CD8s are ameliorated in their phenotype. They downregulate some inflammatory molecules, some inhibitory molecules, and upregulate activatory molecules. In the tissue, what happens is that you can see on the top in this histology in brown are all the hepatocytes in which the virus is replicating. So you can see that the CD8 T cells alone, leukocytes just get stuck around the big vessels inside the liver, and the virus is replicating. But if we put CD4s in the system in the co-transfer condition, then we start seeing inflammatory foci within the liver parenchyma, hepatocytes dying, and also the viral replication is reduced, assessed by histology, southern blot, but also reduced viremia in the blood of those mice. So basically, if we add some CD4 T cell helpers into this system, we're able to sustain a little bit more the CD8 T cell response against the virus in our model. So then we wanted to understand, okay, where is this happening? This was the first question, because we wonder whether this was something that was specific of the liver, or this was maybe a cooperation or an interaction that was happening way before in a secondary lymphoid organ. Then we were seeing this in the liver downstream. Well, to understand this question, we prevented the recirculation of T cells in secondary lymphoid organs. And we did this by splenectomizing the mice. So we removed the spleen and we treated the mice with a blocking antibody, preventing the entrance of T cells and naive T cells in lymph nodes. So basically, in these mice, T cells cannot recirculate in secondary lymphoid organs. Well, what happens in this case? Nothing, because there's still cooperation in the liver, there's still the liver damage, CD8 T cells are still receiving help from CD4s, there's still inflammation and infiltration in the liver, and viral replication is inhibited, meaning that in this case, in our model, CD4s and CD8 don't need to cooperate in secondary lymphoid organs, but they can do that directly in the liver. This next question was, we asked, of course, who is mediating these, because we know, well, we have some exception, but CD4s and CD8 don't really interact directly, so they need an APC as a bridge. And interestingly, we went just after 24 hours after the transfer of these T cells in these mice, so during the priming phase, and we could see, you can see in yellow CD4s and in blue CD8s and in white Kupffer cells, which are the resident macrophages in the liver. We're surprised to see that during the priming phase, we could see in the liver these three cell interaction foci in the liver. So basically, CD4s were jumping from one KC to another, likely undergoing the licensing process. And this pointed to the fact that maybe the APC bridge inside the liver in this context were Kupffer cells. And indeed, if we remove KCs with a very specific model that is called CLEC4F DTR mice, basically, if we give diphtheria toxin to these mice, we conditionally deplete KCs, sparing all other macrophages and dendritic cells in our body. Well, if we take out KCs from the system, nothing happens anymore. There's no liver damage. CD8s are not expanding and not differentiating anymore, and the tissue is completely clean. So basically, this means KCs were the ones mediating the communication between CD4s and CD8. Last part of the project was then dedicated to understand which was the molecular mechanism behind this T cell cooperation in the liver. Well, upstream was kind of straightforward. We discovered that CD4 T ligand is the essential signal that will regulate then the cooperation downstream. So basically, if we block CD4 T ligand, nothing happens anymore. So CD4s will license cases via CD40, CD4 T ligand interaction. Then we checked IL-12, which is a known signal, a known cytokine produced by APCs after the engagement of CD40. But IL-12 gave us an intermediate result. So you can see by the histology that we still have some inflammatory foci, we still have some infiltration, viral replication is a bit inhibited, but basically it's not blocking IL-12 is not completely abolishing this cooperation, but reducing its magnitude. So what we think is that CD4's license KC via CD4 T ligand, and this will trigger the production of IL-12 that probably is sustaining more the response of CD4s than being essential for CD8s. Indeed, the next step was trying to understand what was the next downstream signal. So basically, what are KC doing that is helping CD8 downstream? And this was kind of a surprise, but we discovered that IL-27 is doing the job. Because if we remove IL-27 from the system, nothing happens anymore. The tissue is completely clean. There's no infiltration, no inflammation. CD8s are not expanding anymore, but CD4s don't care. So basically, IL-27 is the factor that is produced by licensed Hooper cells after interacting with CD4s that is then is able to reinvigorate the response of CD8 T cells. And indeed, we asked, can we leverage IL-27 to reinvigorate CD8s? Well, if we now keep CD4s out of the system and we just put CD8s, they're usually dysfunctional CD8s in our HBV transgenic mice, but we give after 1 day a shot of IL-27, we can rescue their function. So we have liver damage, we have more effector CD8s, they produce more interferon gamma, and there's a huge infiltration in the liver and killing of infected hepatocytes. And as a last piece of data, we wanted to understand also if this could be relevant for patients as well. So in collaboration with Antonio Bertoletti's lab in Singapore, we isolated PBMCs from chronic HBV patients, and we put HBV-specific T cells or CMV-specific T cells as a control of memory T cells from these patients. We put them in culture in the presence or absence of IL-27 and HBV peptides. And we saw that after 10 days of culture, the presence of IL-27 was ameliorating the function of HBV-specific CD8s. So to conclude, what I showed you today is that during HPV infection, a peculiar immunological event happens, and it's hepatocellular priming. CD8s are priming the liver, and this renders them dysfunctional. But if we provide them with CD4 T cell help, CD4s will be able to license resident macrophages in the liver via CD4 T ligand. This will trigger the production of two cytokines. IL-12 will sustain the Th1 CD4 T cell response, and IL-27 instead will be useful to reinvigorate the response of dysfunctional CD8s inside the liver. And with this, I'm done. This is Matteo's lab. A lot of people were involved, in particular Christian, Chiara, and Pietro, and a lot of collaborators that I have to thank and I want to acknowledge here. And then even though not directly involved, the new lab members that helped me in this onboarding process here in Paris. And with this, I'm done. I will be happy to take questions.
Thank you so much, Valentina. This is a super interesting presentation. I just like to remind the audience that you can ask questions to our speaker in the Q&A function of Zoom. We have one question from the audience. Is it possible that KC cells promote cytotoxic CD4 T cells? And would it be possible for IL-27 to boost interferon gamma in CD4?
So yeah, it is possible. We looked into the possibility of, we checked for cytotoxic CD4s in our system. So this model that we use, of course, is very somehow artificial. So these CD4s have a single specificity for HBV. So in our case, our CD4s are not acquiring any direct cytotoxic activity. And also we tried to put these CD4s alone in those mice, and we were not able to see any direct cytotoxic activity of CD4 T cells alone. So in our model, it really seems that CD4s are doing something then downstream to regulate the cytotoxicity of CD8s. And then regarding IL-27, it can be. Actually, IL-27 is well known for its effect on CD4s, not really on CD8. That's how it was discovered initially. So it is known IL-27 indeed to sustain the Th1 response in CD4 T cells, in particular helper 1 CD4. So yes, it can sustain both interferon gamma and cytotoxicity also in CD4s.
Interesting. Another question from our audience is, what other molecules on or produced by KCs permit this bridge? Is there any other molecules beyond IL-27? Is there a potential role of STAT3, for example?
Yeah, we don't know. This is something that for sure will be done in Matteo's lab in the future. I think as a follow up, we don't know precisely what is happening to KC. So what I could see is that removing IL-27 was really abolishing everything. So it was kind of striking. We don't know if other cytokines might be involved in the process. I would guess yes, of course, like both upstream with CD4s and downstream with CD8s. So of course, like the whole picture is still to be investigated. But yeah, of course, for sure, STAT3, for instance, absolutely is also downstream IL-27 signaling. So it's something that they're looking at in Matteo's lab.
Another question from our audience is, what's the regulatory loop for limiting the extent of this pathway's activation? Is it mainly IL-35?
Uh, Very interesting questions. We don't know. So actually, one idea that we have is that IL-27 has been seen. So the one of the peculiar thing about IL-27 is that it triggers together with cytotoxicity and effector pathway in CD8s is also triggering IL-10 production by CD8s. So it's a cytokine that alone can, on one hand, trigger an effector response, but also somehow damping the same effector response, producing a cytokine that will somehow control the whole situation, let's say. So this is interesting. It might be that this cytokine signal is balancing the effector potential of CD8s by also making them produce IL-10. So this is one aspect that we think it might be going on in our system.
Great. I guess another question I had was, is the rescued CD8 T cell phenotype durable, or does it depend on like continued CD4 T cell presence?
We don't know. So this we just did a couple of pilot experiments actually for the regions which were really initial experiments, but it was interesting to see that without CD4s, just giving IL-27, CD8 could sustain a medium level of liver damage for many days, like up to 2 weeks. So even in the absence of CD4s, this initial priming in the presence of IL-27 was interesting to see that he's able to keep CD8s on the loop, let's say. So this is interesting. We don't know what does it mean, but it will be investigated in the future for sure.
That's great. Well, thank you so much for a wonderful
Thank
talk,
you.
Valentina.
Thank you very much.